And okay, you ready?
Yes.
Okay.
Okay. Hi, everybody. Thank you for that introduction, Sarah. Like Sarah said, I'm Courtney Murphy, and I did my thesis research on the Puget Lowland Prairies studying golden paintbrush, which is one of the host plants for Taylor's checker spot butterfly, which we just heard a lot about from Alicia. So for the last 2 years, I have been the butterfly program coordinator at Sustainability in Prisons Project. I've been helping facilitate a captive rearing and breeding program for the endangered Taylor's checkerspot butterfly with a group of incarcerated women at Mission Creek Correction Center for Women. It's been a really great job. I've learned a lot. And it led me to be interested in learning more about the Taylor's checkerspot butterfly in the field. So Taylor's checkerspot butterflies, their behavior and life cycle is highly dependent on temperature, precipitation, and other kind of conditions out in the field. I'm not going to go too much into their life cycle because I have a lot to cover. But one important thing to remember about them is that they go through this period called diapause, which is essentially like a 6-month or sometimes around 6-month hibernation period, where they're very inactive, they're not eating anything. And then when they wake up, which is usually between like late January into March, depending on the year, they're very, very hungry and they need a lot of food. They have very highly specific habitat needs and not very many food sources. So here are 3 potential food and oviposition sources for Taylor's checker spots. So right now, like Felicia was saying, they're adapted to use this plant, Plantago lanceolata, or common plantain. And it is a non-native species, but has been in the area for about the past 200 years or so, a little less than that. And checker spots have been adapted to use this species since they were first described back in the late 1800s. But historically, they've relied on native species such as Castilleja hispida, harsh paintbrush, and Castilleja levisecta, golden paintbrush, as well as some other species that are kind of less common. But in addition to checker spots being dependent on variable based on like temperature and precipitation, so are their host plants. So back in 2021, we had a really big heat wave. You might remember it. It was like 110 degrees for a couple days in a row. I didn't enjoy it very much, and neither did the plantain out in the field. So that heat wave really decimated the plantain population back in 2021, and it took it a few years to recover. So that left a lack of plantain for checker spot larvae. So WDFW biologists had to plant extra plantain more than they usually would to try to compensate for this like lack of food in the field in order for us to be able to release our checker spot larvae that we had been raising at the Mission Creek lab. And even with their planting efforts, we weren't able to release all our larvae. There just wasn't enough habitat. So this is a photo of a lot of checker spot pupa that would have been released as larvae that we had to retain for a few extra months in the lab and let them pupate and then release them later to deal with this lack of food resources. So it was a pretty big problem. And experiencing that made me think more about what are checker spots native host plants like, and think that it would be a good idea to learn more about their conditions so that we can diversify what plants we're relying on when we're thinking about releasing checker spot larvae. So I chose to study golden paintbrush specifically. In my study, currently checker spots and golden paintbrush don't coexist in Washington, but we do know that they have utilized golden paintbrush in the past. So up until the 80s, there were observations of checker spots utilizing golden paintbrush. Recent studies show that they're still able to utilize this plant, even if they don't naturally in the field right now. And this plant has a similar phenology to harsh paintbrush, which we do know that they still use at various reintroduction sites. We also know through past research that golden paintbrush, like checker spots, is really it varies a lot based on microsite, and there's a lot more to learn about that. So I chose to study this plant. So this led to my research question, which how does microsite variation affect the phenology, abundance, and growth of Castilleja levisecta as a host plant for Taylor's checker spot butterfly during their larval feeding stages? So I studied this plant throughout basically the entire time that checker spots would be larvae. So from post-diapause emergence in late January, early February, up through the time that they would go into diapause. So I had 3 study sites, and each of these sites represents a habitat where Castilleja has been reintroduced in the last 10 to 20 years or so, Castilleja levisecta. So one of my sites was Glacial Heritage, which many people in this area are familiar with. Glacial Heritage used to have a checkerspot population that was reintroduced and no longer has a population. Another site is Cabinus Ranch nearby, currently doesn't have a checkerspot or has never had a population. And then I had also a site up on Whidbey Island at Pacific Rim Institute. And so these are 3 sites that could be considered for reintroduction efforts in the future. And so it's important to learn more about the host plant phenology at those sites. So how did I determine my microsites? So for this study, I selected microsites like visually based on slope and aspect. Sarah and Adam helped me out with that, so thank you very much. And these sites, or these microsites probably could be categorized in many different ways, but this is what I went with for my study. So my 4 categories were hot, dry, cold, and wet. So hot microsites were flat, there was like no slope, they were totally sun exposed, no shade. The hot, dry microsites were on south-facing slopes of some sort. The cold microsites were either like on north-facing slopes or in areas that could experience some shade, so cooler conditions. And then the wet microsites were like in swales or depressions, places that you would think would have more moisture. So I started my research in late January, and so this is kind of my timeline. So first, I set up my plots. I established my like subsample plant or Castilleja individuals to measure, and then I returned approximately every 2 weeks through July to track percent cover, height, and their rate of senescence toward the end of their life cycle, because I thought those variables would be the most relevant to checker spots. I also had temperature loggers that I installed at each plot to track temperature so that I could see if my predictions of what the temperature of the sites would be like were accurate. And so each of my plots was a meter squared, and I had 5 per microsite at each site. So I had a total of 60 plots. This is what a Castilleja levisecta looks like when it's tiny. I think my dad took that photo, so shout out to him. My parents helped me with some of my research. And so this is an example of kind of what my plots looked like over the course of my study. So the first picture up there, you can see there's not a lot of vegetation, but all of those white things are tiny plants basically. And then by the end, there's a lot of Castilleja as well as other plants like Oregon sunshine. So some of those yellow is Oregon sunshine, not all Castilleja levisecta. This was an example of a very successful plot with like a high percent cover. Not all of my plots turned out to be so luscious. This is an example of a plot at Glacial that ended up actually growing no Castilleja, despite the fact that there was the presence of like prior year stems. And this was one of the hot microsites in my study, and you can tell like it was very sun exposed and really didn't grow well at all. And I actually used an alternate plot to track senescence because I couldn't track senescence if there was no plant. But you can see how some of the plots changed a lot over the season and some really did not. And to track percent senesced, I came up with this like rating system where I tracked qualities like discoloration of like leaves and leaf bracts, like desiccation of leaves, and like production of seed pods, so that I could kind of go back and consistently track percent senesced. So what did I find? Whoa, this one. Oh, right. What did I find? So I found that temperature didn't vary that much between microsites, but you can see that overall, especially in terms of average lows, the hot sites were on the hotter side, and then the cold sites, blue, were always colder. So that was, good to find. And I'm not going to talk too much about that, but because I don't have that much time. But average highs on top, average total, and then average lows on the bottom. Species richness. So I also did a species richness assessment to kind of characterize my microsites. And so something I found interesting through here is that there is a pretty consistent pattern of species richness throughout the microsite types. We have an outlier here at PRI where the hot microsites had a higher species richness, which was atypical compared to the other ones. However, as you could see in the temperature graph up here, Pacific Rim just had lower temperatures overall, so the hot sites there were not experiencing the same level of heat, and that could explain some of the difference in species richness. But overall, you can see that the like colder and wetter sites had a higher species richness overall than drier and hotter sites. So you can see through this that my sites really were distinct from each other. In terms of percent cover, I found that each site differed in terms of which microsites had the best Castilleja levisecta growing conditions. The shaded areas on this plot represent times where there would be checker spot larvae active, so you can kind of see like what the post-plant availability might be while larvae are out there. And so glacial heritage really had no significant difference in percent cover. The Castilleja levisecta population was just so low at that site that we couldn't really determine significant difference. But at Cavness Ranch, there was significant difference between microsites, as well as at Pacific Rim. And at Pacific Rim, there was also a significant difference with interaction between microsite and time. So you can see that the wet sites at this microsite really accelerated in terms of their percent cover growth rate. And so you could imagine that microsite conditions were affecting that. But I felt like this research really highlighted that each site does need to be evaluated individually. It's kind of hard to, I mean, I didn't find a consistent trend between sites because even though the microsite types were similar, like the topography and other conditions at each site was so different. In terms of height, I found that microsite, at least the way that I categorized it in my study, did not have a significant effect on height of Castilleja. Except at Pacific Rim, the cold sites, which you might have remembered had the lowest percent cover, had the highest height. And I think that in this case, that had to do with the fact that those sites had kind of a different plant community than other sites. Those sites were kind of closer to a forest edge and had some plants you might not usually find on a prairie, such as Oregon grape and bracken fern. And so it seemed to me like that was really limiting the spread of Castilleja levisecta, but the levisecta that was able to persist ended up taller because it was trying to outgrow the other plants in the area. So I think that that's what explains that difference. And then in terms of senescence, I found that at Cavness Ranch and really at all sites, there was a significant difference between the microsites in terms of their rate of senescence, in terms of how their, I guess, rate and completeness of senescence by the end of the study. And specifically at PRI, well, at all sites, you can see that there's a trend of the hot microsites senescing faster because they are more exposed. I also encountered something that was kind of challenging in terms of tracking senescence is sometimes plants would get browsed or they would have never actually grown big enough and flowered. And so they would senesce at different rates than plants that had completed their full flowering life cycle. So that's something that also may come into effect here. Got it. Yeah, so that is what I found there. And then, yeah, so summary of my results. Like I said, species richness definitely differed between microsites, with the trend of cold and wet microsites having a higher species richness than others, except for at PRI, for reasons I already discussed. I found that the effect of microsite on percent cover was not consistent between sites. It was greatest in the wet microsite at PRI, the hot microsite at Cavness Ranch, and had no significant effect at glacial heritage. Microsite did not affect height except at PRI, and the cause may have been species composition in those plots. And microsite did have an effect on percent senesced at all sites, and there's a trend of hot microsite senescing faster and more completely, at least during this study. So what does this mean for the future? Like I've stated, microsites were distinct in the study, but were inconsistent between sites, making comparison between those sites difficult. I think that future studies could really benefit from a year-round temperature monitoring prior to, temperature monitoring prior to actually collecting data on plant growth. Even though I did find that there were, appropriate differences between temperatures of my microsite, I may have chosen differently if I had been able to monitor ahead of time. And also things like monitoring of soil moisture and testing soil types prior to tracking your plant species would be really informative. I would have loved to have been able to do that if I had a whole nother year to do my study, but I didn't. And also, a longer study time There's so much variation year to year, and you'd probably be able to see more consistent trends if you were able to track multiple years in a row, which is in the works. So it will happen. And I found through just my time out in the field and also being able to compare that with what I know the checker spots are doing from my work with SPP, that there were multiple periods of overlap between when Castilleja levisecta would have been available to checker spots and when checker spot larvae were active. And so while it's true that it doesn't emerge as early as plantain because plantain overwinters with its leaves, if checker spots were out on these sites, they would have been able to utilize Castilleja as a food source and also probably as an oviposition host plant. So that suggests that it could be utilized as a secondary host plant in addition to plantain. That's my presentation. Thank you very much, everybody. And I would love to take questions.